Imagine a machine that can read the code of life, not in centuries, not in years, but in weeks. Picture a world where labs across the globe, classrooms, and even curious hobbyists share the same tools and tricks to read and rewrite DNA. That change is not theoretical, it has already happened, and it bends the future in ways both dazzling and alarming. By the end of this video, you'll understand exactly how we went from a $3 billion government project to DNA editing in your garage, and why that transformation might be the most important story of our time. The story begins with a feat that once seemed impossible, mapping the human genome. What in the 1990s required governments, hundreds of scientists, and roughly a decade was an enormous and costly undertaking, a project commonly summarized as costing about $3 billion and concluding after 2020. What followed, quietly and relentlessly, was an explosion of automation and innovation. The grind of turning DNA into digital information, which used to be slow and specialist work, has become fast and increasingly cheap. Within a decade and a half, the price of reading an entire human genome collapsed from millions to roughly the cost of a small car, putting enormous power in far more hands than anyone predicted. Those numbers matter because they are the engine of a deeper change. Biotechnology has left the protected lab. Today, we use engineered organisms to make insulin, to create crops that resist drought, and to connect prosthetic limbs to the nervous system in ways that restore movement and dignity. These are not sci-fi promises. They are real, life-saving advances already in widespread use. At the same time, the same openness that shares life-saving techniques also spreads the instructions and tools that could be misused. The line between a medical breakthrough and a dangerous application can be thin when the knowledge and the parts are affordable and available. You know the last global pandemic, the virus that changed everything. The official reported death toll runs into the millions, a grim marker of how fast a microscopic agent can sweep the planet and how unprepared systems can be. That pandemic is a blunt example of two facts. Microbes spread fast, and human systems react after the worst has already happened. What makes the present moment different is not just natural spillover from animals or bad luck. It is the combination of widespread biological knowledge, digital databases of genetic blueprints, and commercial services that can synthesize DNA. Services that, in some cases, have been shown to present biosecurity vulnerabilities when orders are not properly screened. So how did we get from a handful of elite labs to this near-universal toolkit? The answer is twofold. First, technology followed a path of rapid cost decline. Machines that once filled rooms became square-foot lab benches, then desktop devices, and now cloud-connected services. Second, knowledge flowed outward. Papers, open databases, online protocols, and community labs accelerated the spread of techniques. Where it once took years for a trick to travel between institutions, today it often takes months, and within a few years, the same techniques show up in undergraduate labs and community biology spaces. This democratization is a force for creativity, but it also multiplies points of failure. That combination of cheap tools and shared information creates a troubling possibility. Genetic sequences, the exact instructions for viruses and bacteria, are routinely shared in scientific journals and public databases because openness accelerates discovery. Yet those very sequences can act as info hazards, instructions that are dangerous if misapplied. Add to that a market where DNA strands can be ordered and assembled, and the risk landscape changes from theoretical to practical. Experts and analysts have pointed out that without careful safeguards, the infrastructure of modern biology lowers the barrier for someone to attempt crafting dangerous agents. What would a future of misuse look like? It doesn't require a Hollywood villain or a secret government program. It could begin with an earnest scientist chasing publication, a hobbyist trying an experiment that goes wrong, or a bad actor leveraging commercial DNA services. We must be careful here. I will not describe methods or how-to steps because those are the kinds of details that should never be shared. What matters instead is understanding the dynamics, ease of access, the public sharing of sensitive data, and the limited screening and oversight in parts of the supply chain that can let small mistakes cascade into global crises. Are we helpless in the face of such a future? Far from it. The story of the atomic age offers useful lessons. Powerful technologies demand governance, tracking, and transparency. In the world of biology, experts have proposed concrete layers of defense. First, we can treat certain genetic data as sensitive and require stronger controls or screening before sequences or synthetic orders are fulfilled. Second, we can build better detection systems, environmental monitoring and rapid genomic surveillance that spot unusual patterns in air, wastewater, or clinical samples before they bloom into full crises. And third, we can accelerate defensive measures, faster vaccine platforms, pre-approved antiviral libraries, and physical technologies that reduce transmission in public spaces. None of these is a silver bullet, but together they change the odds. Recent analyses and policy proposals emphasise this three-part approach as the backbone of future biosecurity efforts. There are already practical steps underway. Some DNA synthesis companies screen orders against lists of dangerous sequences and verify customers. Regulators in several countries are moving to tighten standards. Researchers and policy groups are designing better surveillance networks that blend biological sensors with machine learning to spot anomalies early. And scientists are working on non-pharmaceutical defenses, from air filtration systems that can trap virus particles to ultraviolet systems that inactivate pathogens, which can blunt outbreaks even while vaccines are being developed. These are the tools of resilience, not glamorous but essential. Pause here for a moment and consider what this implies for each of us. Biotechnology does not live in a vacuum. It is embedded in economies, classrooms, startups, and global supply chains. The choices we make, from funding rapid diagnostics to insisting that commercial providers screen orders, will shape which side of the ledger wins. The one of miracles or the one of avoidable disaster. If you care about that balance, small civic actions matter. Support transparent science, demand sensible regulation that protects safety without killing innovation, and encourage investment in detection and rapid response. If this feels urgent and you want more deep dives into how science is reshaping our world, a simple thing helps me keep making episodes like this. Please like the video and subscribe. It only takes a second and it lets us keep exploring the big questions together. When you step back, the picture is both unsettling and hopeful. We have unlocked a power that can end suffering, curing genetic diseases, growing food where none would grow before, even restoring lost functions after injury. At the same time, the same knowledge creates vulnerabilities we must treat with seriousness and imagination. The future will not be written by technology alone. It will be written by the institutions, laws, and cultures we build around that technology. So where does that leave us tonight? With a choice. We can treat biology like a toolbox to be shared without thought, or we can steward it, making it available to heal while erecting smart barriers against harm. That stewardship is not anti-science. It is a mature way to love science. It asks for responsibility, not retreat. It asks for vigilance, not fear. If this resonates and you want more thoughtful looks at the crossroads of science and society, like the video and subscribe for more. Together, we can keep asking the curious questions, learn the complicated answers, and face the future with both wonder and clear eyes.